and found to be quite different and had a much reduced healthy diet index compared to the community dwellers, whereas the community dwellers had a larger intake of fruits and vegetables and a more varied diet, whereas it was much more monotonous in the case of the long-stay individuals. So, moving on then to microbiome diet, dietary interventions that might positively impact the microbiome in elderly in particular. So this is just a summary of, am I okay for 5 minutes? Sorry. Okay, the New Age study looked at the impact of 1 year dietary intervention in persons over the age of 65 in different countries in Europe, and demonstrated that Mediterranean diet intake for 12 months led to a positive impact on gut microbiome, with a number of diet positive taxa and a number of diet negative taxa described as a result of the dietary intervention. So the Mediterranean diet led to enhanced microbiome composition and function, reduced inflammation, reduced frailty in the subjects, and enhanced anti-inflammatory activity, enhanced cognition, and improved grip strength, demonstrating that the Mediterranean diet not only impacts on microbiome composition and function, but has significant impact on enhancing the health of the consumers. Another way to restore the gut microbiome in early life could be to develop consortia of microbes. And again, this study by the O'Toole group led to a selection of a large range of microbes from the elderly gut diet, from the elderly gut, characterized the strains and blended them into a microbiome consortium of 100 different species that actually were shown in an ex vivo study to restore the gut microbiota composition and function in an artificial colon model. So I suppose I've told you that diversity is the spice of life. Enhanced diversity of the diet leads to a good microbial diversity, and on the flip side, a reduced diversity in the diet is also associated with negative health consequences for the host, but also for the microbiome, leads to enhanced inflammation and enhanced frailty. So, diversity is the spice of life. And I've told you that the microbiome has intimate ties with many facets of host health, integrally involved in intestine, adipose tissue, cardiovascular, metabolism, etc. And just to say that non-communicable diseases are on the rise and cause a number of problems for human health, including reduced morbidity, dysfunction, and quality of life. And microbiome alterations associated with non-communicable diseases can potentially be corrected via modulation of the gut microbiome via various dietary approaches and therapeutic interventions. And one area that's well recognized now is the obese diabetic microbiota associated with an altered microbiota composition with negative consequences for the host, involving low-grade inflammation, which leads to all sorts of problems in various organs of the host. And also gut-brain axis, I mentioned it before. It also is associated with potentially, the gut microbiota can negatively impact on the brain health and can lead to phases of depression. And so I just want to kind of finish 2 minutes with dietary fiber and dietary diversity can impact microbiome composition and diversity, leading to an enhanced microbiome for an enhanced human health. This study just describes an overview of microbiome in US individuals versus Papua New Guineans who consume a diet rich in fiber in comparison to Americans who consume typically a diet more rich in, or more devoid of fibre, and the Papua New Guineans demonstrated a higher alpha diversity, higher levels of Prevotella and bacteria associated with high quality gut microbiome. And so therapeutic modulation of the gut microbiome via precision microbiome modulation is an opportunity to enhance the microbiome composition and enhance the health of the host going forward. So my last slide, sorry. I think I'm okay. Feeding the microbiome throughout life. Human microbiome is inseparable from host health. And from the beginning, a good start with a natural birth, breastfeeding by a well-nourished mother and avoiding antibiotics is the best way to seed the early life microbiome for optimum health and development. Then maintenance of a high-quality gut microbiome can be achieved via avoiding junk food diets, taking adequate dietary fibre, adequate exercise, and a diverse diet throughout life. Modern dietary practices of low dietary fibre, i.e. the Western diets, have been linked to an increased prevalence of non-communicable diseases, for instance, gut diseases, such as inflammatory bowel disease, obesity, type 2 diabetes, and metabolic syndrome. Dietary fibre consumption is necessary to feed our gut bacteria. It promotes microbiome diversity and functionality, enhances microbial short-chain fatty acid production, ensuring optimal activity of this virtual organ. So thank you very much. I just want to acknowledge the people on this slide who contributed to some of the results that I shared.
Thank you very much, Catherine, for this wonderful overview of the development of the microbiota from birth to older age. I would suggest that we will continue now with the presentation of Christophe Lacroix, and then that we will discuss the two presentations before the coffee break.
Perfect. Thank you very much.
Thank you. So I will now briefly introduce Christophe Lacroix. Most of you, of course, will know him, but I'm sure that the people that are following us online, not all will know him very well. Well, Christophe Lacroix is full professor for food biotechnology at the Institute of Food Science and Nutrition of the ETH Zurich. Additionally, he's the founder and member of the board of directors of the ETH spin-off company PharmaBiome, which is specializing in the design and production of gut microbe-based products for human therapy. I think that's very important. If we have listened to Catherine Stanton, I think it's a very important approach. His main research focuses are development of technologies for production of lactic acid and probiotic bacteria, molecular characterization and utilization of specific metabolic activities of food bacteria, study of the intestinal microbiota with a new in vitro colonic system with immobilized cell that he has developed and which is very useful for this in vitro research. He, of course, has also published a lot of papers. He has a Hirsch index of 80 and he has been cited 20,000 times. So, Christophe, it's yours.
Okay, so good afternoon. Maybe it's time to jump a bit on your seat, or if you want to stand for a few seconds. It's a long time, two presentation of 50 minutes. Do you hear me well, yeah, or it's fine? Online, I hope you can also have a good sound. Thank you very much, Christian, for your nice introduction. So it's a pleasure for me today to give a bit of a story. So I will talk about the story of the collaboration between the ETH and the Laboratory of Nutrition and Gastroenterology of Christian at the KISP. To give an idea on the techniques, on the method, on the need to assemble methods as well, to better understand the microbes, not only by their names, but mainly by their function. So I will talk about infant microbiota. I'll start from a long time ago, 2007 or 2008. I made a mistake in my date. It's a long time ago. And I will show you how we approach a mechanism for developing microbes for treating infant colics. So as Christian said, I'm not a gut microbiota specialist as such. I'm an engineer. So but engineer working for now more than 40 years in biotechnology. So working with microbes. I love microbes. I love to cultivate microbes, develop technology, make them more resistant and more efficient when we want to use them in food or in health products. So we need for that to understand the metabolic activities. We need to understand their functions. In particular, in the area of gut microbiota, and we work with the human as well as with animal, mainly chicken and also pig microbiota. Also working very closely with model system. I won't talk about this model system because they become much more engineering. If you are interested, you can read in our publication. But we want to understand how the microbes are responding to environmental factor and to diet. As a university, very much involved in teaching and research, and last but not least, also as an engineer interested to translate the activity that we do in the labs to the application. So in the area of food biotech, there is a lot of transfer. Not gut microbiota, it's a bit more difficult, but functional microbes in food, in fermentation, in allowing to provide functionalities like safety and preservation is an essential goal in this. So very shortly to talk about our program, work a lot on ecosystem, understanding ecosystem, food, environment, and gut ecosystem. And why do we do that? First, to understand what is present, who is doing what, but also mainly to build a culture collection. So we have a culture collection of more than 8,000 tight microbes that we screened from different environments, and from this we can develop functional application of those microbes. So on the slides in the middle, you can see our activity in molecular characterization and physiology of microbes, mainly in the area of antimicrobial produced by microbes in food. Why not in the gut? Carbohydrate metabolism, response to micronutrients, stress adaptation, important for technology, as well as antibiotic resistance. Then the next step, when we have microbe, we need to produce them. So this is my heart. My heart is technology, it's fermentation, it's propagation, it's downstream processing, and it's keeping the microbe until they are used for whatever purposes. So we develop processes, technologies for a food microbe, but very much also for live biotherapeutics. So we are quite advanced in the area, in particular of consortium cultivation, because consortium, the name is simple, the application is extremely complex. Because when you have a consortium of several, sometimes more than 100 microbes, it's extremely difficult to produce 100 microbes, especially gut microbes, and then mix them afterwards in a standardized product to deliver. So there is a big challenge in the technology. And finally, we work on gut microbiota and health, and this is in connection with specialists like Christian. We work a lot very closely with the gastroenterology department of the university hospital. There we work with, we apply our knowledge in microbiology. We are not doctors, obviously, so we develop models. We try to understand the microbes. We try to understand how they respond to different factors. I mentioned probiotics, antimicrobials. Micronutrients is a big area of activity, especially in relation to Africa, where we need to supply micronutrients to infants. And I've been working for the last 12 years on how to best formulate the micronutrients to prevent the bloom of enteropathogens in collaboration with the Human Nutrition Laboratory at the ETH in infant, because iron, we must give iron to infant at 6 months. Most of them are anemic, and very well known that iron is not is promoting essentially enteropathogens. So big issues with this, and yeah, when we develop microbes for application in health, obviously we want to understand the mechanism of actions. So we have different approach for this, where we range from in vitro to in vivo, but again with the multidisciplinary approach and trying to assemble the competencies around the project and covering from the molecular characteristics of the microbes until human studies, again in collaboration and supported by different grants and also quite a bit by companies. So in this area, there are different scale of investigation. There are also different methods to investigate. So we hear a lot about molecular methods that are essential. But to my view, humble view, I don't think they are sufficient. We need to have also the real microbe. We need to read the functionality. We need to be able to cultivate, to read the metabolites, to combine the microbes, to produce consortia, and eventually to see the ecosystem at work. When we take fecal microbiota, but they are not at work. They have already transited through the colon, so the main activity open in the proximal colon. Yeah, we cannot get samples from there. And in between, until the microbes comes out, there is absorption, there is exchange with the host, there is transformation of the metabolite. So it's important to read feces, but it's a proxy. It's only a proxy. So many of the methods that are used in microbiome, they are proxy, but they are excellent methods, and we need to reconstruct them to come to mechanism and to understanding. So that's what I've extracted an area of application which was done in collaboration with Christian. So infant gut microbiota and functionality, and I wrote 2007, maybe 2008. I thought it was before, so we work with different doctoral students, 2 postdoctorates, 7 peer-reviewed scientific papers, and two patents. One has been granted. So in this collaboration, we first work with microbes as antimicrobial. So I won't talk about it. Then we switch to milk microbiota. And this is where we started collaboration in order to understand the very early microbiota of the infant and how milk could contribute. Is there a real translocation and supply of microbes from the mother's gut to the infant? It's not easy to demonstrate. You might find the 2 microbes in the 2 systems, but how can you be sure that the microbes come from the milk? It can come just from fecal contamination. It can come. So we wanted to try to find or to provide some support about this entero-mammary pathway of microbe transmission. Then from this we developed, we looked a bit at the composition of the milk microbiota, and we saw functionality profiling of the microbiota in the milk, which are very consistent with what we find in the infant. We find functional groups, and on this we built another research which essentially looked at the microbial development, but this time based on function. And this study, we did it over a 24-month period to see the evolution of the microbe, but also of the function in the system, with the main focus on lactate. It's a very important metabolite, lactate. It has to be removed because it's toxic. Yeah? And for that, you need microbes which produce lactate. There are plenty. The gut microbiota of the infant is mainly lactic acid producers. But what about the lactic acid utilizer? They need to be there. We need to understand them. And why not? Maybe some pathology are related to this balance between both groups. So I will talk very shortly about this with the example of colics. So we tried to address this issue with trying to understand, is there a functional, colics, it's not easy. They happen with all kind of infants. We don't see real microbiota pattern. There are different products on the market against colics, but it's a microbe which is used, no mechanism. There is not a single mechanism on colics. So it's very difficult whether Lactobacillus reuteri, GG, or whatever bifidobacterium works, because it's very difficult to demonstrate if it's active or not against colic, which is very short time and for which you don't have really a precise profile, for microbiota. So we try to work on this, but looking at the function. Is there something wrong in the function? Is there lactate accumulation? Is there lactate utilization that might explain that the infant become bloated, is in pain, is crying? And this is quite an important stress for the new parents. I had my first kid at colic until 6 months. I tell you, it's quite tough. It's tough when it's for 6 months. So I was motivated by my personal activity as well. And then the very last project that I won't talk about it, that we are currently finishing, indeed, is about FAPD NOS. So I wrote the full name. It's functional abdominal pain disease in pediatrics, not otherwise specified. So I won't say more about that, but just in order to try to relate this pain in pediatrics, or in not infant, it's more in toddler or later in life, and for which there is no cause, no reason for that. And what we tried also to apply is the same approach, a cohort study done in Switzerland and in Croatia, if I'm not wrong, and trying to read the microbiota in terms of function. It's not easy. We don't see much in terms of profile of the microbiota in this disease as well. And we tried also to relate with early stress in life. Because we had an ETH professor, Isabelle Mansoui, who is a specialist of early stress and behavior, and how this can be communicated to the descendants over many generations. So we formed a group on this. So I don't want to spend too much time. I won't repeat what Catherine presented very, very nicely about the initial colonization, the factors. Yeah, there is a dogma for the colonization of the infant. It starts with aerobic conditions, so it means that at the beginning we have a bloom of enterobacteria. Then, as soon as the condition becomes more anaerobic, then there is a development of bifidobacteria, and Lacnospiraceae. Until you reach a certain maturity with increased diversity, which is observed after 2 to 3 years of age with adult type microbiota. So this is kind of a dogma. We wanted also to understand a bit better the early stage. When does it happen, this anaerobiosis? Is it like it was suspected 1 month, or it has been proposed 1 month? Well, I hope not, because the poor anaerobe, they would not be too happy. So it might happen very quick, and we know microbes are able to very quickly remove oxygen of the system. So we looked a bit more precise on the very early period of age, and we looked also at the functions and just to illustrate why functions. Yeah, essentially, this is the human microbiome project cohort study, and you can see on the it's on your left hand, I think, too. The profile, it's 242 healthy individuals. You have the profile of the microbiome at the phyla level. You can already see that there is a big dispersion of the colors. Just think about the main phyla. On the right, the functions. So you can see that even though you have a tremendous difference in the composition, the names, and this is phylum. If we go down to families, we go to genera and so on, to species, but there is even more diversity. But we see that the functions are kept. So when a microbiome has a composition and another one is individual, has a complete different composition, doesn't mean they don't do the exact same function for the host. And we hope so, because a healthy microbiome should deliver the same function to all individuals. So this is I think, an important also output of this paper, where function is maintained, whereas phylogenetic diversity is great. So the microbes, we heard a lot about diet, and we will hear also later in this afternoon about the impact of diet. Yeah, the diet is feeding the host. It's the first thing when we design food. We try to calculate the protein, the sugar, the vitamin, but essentially to fulfill the host. But the host is not the only one to feed. The microbes are also eating part of the diet. And while doing so, they can grow, they can establish a microbiome, they can produce a range of metabolites up to more than 500,000 metabolites. So we are in the superlative with the microbiome all the time. We know only a few. But these metabolites are afterwards absorbed or are interacting with the host, and through this metabolic activity, the microbes contribute to the development of the infant, contribute to the maturation of the immune system, to the colonization resistance, and to the digestive capacity. So the metabolites, microbes are important, but the metabolites are also very important to understand in a microbiota ecosystem. Okay, so we know that there is a trilogy here where the diet is impacting on the microbiota, the host factor is also impacting on the microbiota, and the microbiota produce metabolites that feed the host or exchange with the host. And we know also that when there are perturbations, this might be the cause of many diseases and disorders. So it's very important to understand the role of the diet, but not only the diet, but specific components of the diet, because microbes will only see what is not digested, and they will respond very differently to different components of the food. So it's a very big challenge as well to with the complex food, to understand how the food is acting on the microbiome. Because again, it's another level of complexity. We know about carbohydrate, we know more and more about fibers, but obviously the diet has a tremendous impact, not only in the adulthood, but also in the neonatal period, like Dr. Stanton just mentioned before. Okay. Big talker, so I have to be careful. So the point I want to make as well is related to the methods. So there are different methods to investigate microbiotas. So there are the central one, the three big line. They are culture independent. So culture independent, you have the DNA-based methods. There are several. You can use a quantitative DNA-based method like PCR, polymerase chain reaction, quantitative. And you can measure certain groups that are targeted by the amplification. You have also sequencing based on the 16S DNA, ribosomal DNA. And this will give you, they require also an amplification with a special primers, so they will allow to essentially amplify sections of the genes, regions, that are different among different microbial groups in the gut microbiota. And so you need to select the region. So if one study uses a region, the other one another region, you get sometimes it's very different data. So again, it's part of reading literature. It's not easy to read the method. Then metagenomics, yeah, you don't have PCR. You can read the whole genome together, but you need to assemble. So it's a lot of bioinformatics, and you can have also biases in this. They are all excellent methods, but everyone has a bias. So this also partly explains sometimes that studies are not reconciliating indeed. Then you have the transcriptomics, and it will be talked afterwards, I guess, by a speaker. And the proteomics approach, I don't want to say too much. You have the chemical characterization with metabolite analysis, also with metabolomics, but 500,000 metabolites. If you don't have a real question, a real target, very difficult to use metabolomics as a screening tool indeed. And then the culture-based. This is the one I like a lot, but certainly not the only one. Very far from that. We need to have viable microbes. So we use them for enumeration and the condition where we can read functions. So this is the functional characterization as well. We can isolate microbes. We can read their activity. We can do phenotyping, genotyping. So all these are complementary. Maybe you don't need all method. I would say, yeah, chemical are needed, molecular are needed, culture are very much needed as well, not so much applied. But they rely on viability. So what does it mean, viability? By the microbe, they need to be alive. Molecular method, you don't need that. You need to keep the DNA composition of your sample. So you can kill, and you know that gut microbes, they are super sensitive, they are anaerobic, they are sensitive to environmental conditions. So when we start to work with culture, it's extremely difficult. The microbiological techniques are indeed very, very challenging to implement. So I will try to illustrate a bit the use of these different levels for the understanding of the human milk. That was the first study we did, and Maria Carmen after me will talk a lot about this. So I don't want to say too much, except there are microbes in the milk. There is an entero-mammary pathway which is presumed. I think it's difficult to demonstrate, but we can support. That's what we wanted to do. It's a long time. It's a study that we have started 14 years ago. So it was really at the beginning when we started. And so it means whether milk can contribute to infant microbiome development. And I don't want to talk about this. The mechanism, it will be, I'm sure, talked afterwards. So what did we do? We started a cohort. So you see the cohort, 7 healthy mother neonates. It's very small. It took us 4 years to do this cohort. Why? Because you need to recruit the children. We focused on the first month of life. We had 21 started mother neonate pair, but we work with microbiology. So we luckily had the study nurse at hand, with Rebecca, who is in the room today in particular, and afterward Vanessa, it was another study, right after, I will talk about it. So we looked at the microbiome in the mother before birth and in the infant at 3 time points, first, second, and fourth week of life. Looked at feces of the mother and the baby, as well as the breast milk. So we did culture-dependent and culture-independent methods. For culture-dependent, we tried to detect the major population of the gut microbiota of infant. So we used 9 medium. We need to cultivate under anaerobiosis in special techniques. It's called engage. So it's not on plates. It's not the classical microbiology. Even under anaerobic chamber, you cannot grow most of the microbe of the gut under these conditions. So and we isolated strain, and we wanted to try to detect the same strain in the mother's feces, in the infant feces and in the breast milk. You can tell me, yeah, but you have cross-contamination. So we looked to decrease the risk of cross-contamination. We looked at strict anaerobe, so hoping that they would not be given back by the babies when drinking the milk. So it was the challenge of this study. So what we observed in this study, so again, the first paper came in 2012, so it's long now. The science has built a lot in this area as well. We found only in 7 babies 2 very, very distinct profiles. One dominated by bifidobacteria, and this is on PCR, so it's quantitative, not sequencing. Eh? And the other one, there are 3 infants, they had very, very low bacteroides and non-detectable apart from the very last time of analysis. So very big difference. They were all healthy, breastfed, vaginally delivered, and with no single problem at all. What we also found, it's well known, there is no butyrate producers in infant. Infant microbiota, it's acetate and it's propionate as final product, short-chain fatty acids. So no butyrate neither in this microbiota. And most important, we found that anaerobe, during the first sampling period, 3 to 6 days, they were already 60-fold more than the anaerobe, were 60-fold more than the aerobic bacteria. And we know that we didn't overestimate the anaerobic bacteria because they're very difficult to cultivate. We might overestimate the aerobic bacteria in this range. So it seems that it happens very quickly, this switch from aerobe to anaerobe in the early gut microbiota. And we showed that also by culture and by incubation in different aerobiosis conditions. Okay, so next, so this is by culture. Next, pyrosequencing. So essentially, the pyrosequencing confirmed the fact that the switch occur very early. We can see this in the bifidobacteria, also the bacteroides established very early. What we noticed also, it's on the right hand of the graph. I put in green the lactate-utilizing bacteria. As I explained you, we were very interested. Most of the microbe of the early infant, they produce lactate. So they produce lactate. This is the main metabolic intermediate which need to be afterward transformed. So afterwards, we looked at the breast milk to try to find microbes in breast milk. So we observed a viability of about 10 to the 3. With PCR, we get maybe to 10 to the 6, but because by culture, we cannot recover all the microbes. Now, obviously not. There was also a dominance of facultative anaerobes that you can see, I hope, in this table, that Streptococca, Staphylococca, but they represent the dominant. We didn't find much bifidobacterium in the mother's milk. So we had more lactobacilli, but not many, as you can see. And essentially, the only strict anaerobe of the system, we found proteobacteria, very low level, where the only strict anaerobe were bifidobacterium. It was Bifidobacterium breve that we were able to isolate from the culture. Okay, what we also observed is shown in yellow. Are the lactate-utilizing bacteria, which are very prominent, the Propionibacterium and the Veillonella. And there are two types that can use this lactate. So I will come back to the Propionibacteria, because now they are not anymore called Propionibacteria. They have been reclassified in 2 types, because Propionibacteria, they are the bacteria that we use to produce emmental cheese. So it's Propionibacterium freudenreichi or whatever, Shermani. And you have the other bacteria which are pathogens. So they are the skin Propionibacteria. And those ones have been reclassified as Cutibacterium, whereas the other one are kept as Propionibacteria. So this kind of reclassification makes a big difficulty to read data as well, because we found only the cutaneous Propionibacteria in the infant as well as in the milk of the infant. Okay, when we do pyrosequencing, we observe quite a bit of diversity, but again, we have to be careful because the levels are very low, and we can introduce also noise in the data. You know, when you work with 10 to the 4, 10 to the 6 microbes, you are really at the very limit of pyrosequencing or of high-throughput sequencing. At the time, it was pyrosequencing. And it's very easy to contaminate with reactant, with whatever, and then you find all kinds of brain. But what we observed, we tried to do it the best we could. We observed that indeed there was quite a bit of richness, and this is in the breast milk. Even the richness, again, based on the method, was higher apparently than in the infant feces. It's not surprising because infant feces were dominated by bifidobacteria or bacteroides, so there is not much space in order to detect other microbes. Whereas in breast milk, it's a more equal number, so we can go much deeper in the reading of the ecosystem. Okay, so what we found also in breast milk is that we have a balance between lactate producers. You can read in these slides. Many are known. The Bifidobacterium, Lactobacillus, Pediococcus, Staphylococcus, and so on. And lactate utilizer, there were very few. And very few. By all techniques, we found Propionibacteria, Actinobacteria, and 2 Eubacterium and Veillonella. They were the only 2. So it put us into the track. What essentially is it contributing to? And we looked afterwards on the infant microbiota. We also found in the infant microbiota, so diversity of microbes, but what I want to say here in this slide is we tried to find microbes that were in the free ecosystem, the feces, as well as in the breast milk. And we found indeed a number of microbes, Bifidobacteria, Collinsella. This is based on molecular method. It's not based on culture, bacteroides and so on, that we could find in all 3 ecosystems. Are they the same or not? That was another issue. So we continued with the isolated strain by culture. I mentioned it before, and we tried to find the exact same strain. So we had to go to another level of molecular characterization, which allowed to distinguish strains. So it was a very tremendous work to do on each of the strain and to compare the profile of at strain level in order to try to find the same strain. So we could identify strains shared between 2 ecosystems. With 3 ecosystems, essentially, we found only Bifidobacterium breve that we could show strict anaerobe shared in the free ecosystem. Okay, so here is the trophic chain that we relied on for the lactate. You have here essentially the main microbes in infant microbiota, mainly lactate producers. You can see the list under as well, and digesting remaining lactose and HMOs, human milk oligosaccharides, and accumulating lactate, and then the lactate utilizers, which are shown in this graph, with propionate being one of important metabolites. Butyrate, it's almost absent in infants, so a very, very limited range. Acetate is an important product of bacteroides, which are present. And also, lactate can be used by what we call sulfate-reducing bacteria, and desulfibrio as well. And this, we don't like them. We find them in pathology situation, but we find them as well in the infant gut. I will show you in the next study. So lactate, but it's not a good intermediate metabolites. Sulfate-reducing bacteria, they have been associated with pain, with toxicity. Hydrogen, which is produced also by some lactate utilizer, is creating bloating. So we started from this aspect and presented a conceptual patent without demonstration. So in this patent, indeed, we we reported composition, especially baby food composition comprising living lactate utilizing propionic acid producing bacteria, because those ones, they don't produce H2, but they disseminate very efficiently lactate, in combination with lactic acid producing bacteria to make sure that there is a trophic chain from sugars to propionate as well. And this composition was aimed to treat or to prevent infantile colic, intestinal discomfort, sensitivity, and cramp. So that was, again, a conceptual pattern that can be developed. When you understand a bit the function and the metabolism, you can put it down, but you need afterwards to demonstrate. So this is the second study, quite advanced as well. So this time we worked with, again, with a cohort with 32 infants, 8 colic infants. It's not big cohort, very difficult, again, to work with big cohort. It took several years, I don't know how many, 3 years at least, to perform. Again, because we use a cultivation method. Infant, finding colic infant, it's not that easy, because you need to have the infant from the very beginning as well. So we started with infant at birth, and we had to find colic developing over time in this cohort of 40 infants. So we work with, again, with culture, we work with a molecular method, metabolic method. What we observed is, it's well known, and Catherine also mentioned it, it's chaotic. During the first 6 months of life, very chaotic, very individual development. We had also cesarean and vaginal birth. A very chaotic system during the first 6 months of life. When we go to 24 months, we can start to see more stability and more similarity at the genus level. So I don't want to say too much. Then we looked at the functional group. So by culture and by qPCR. So what we found is the lactate utilizing bacteria, and this was kind of unexpected as well, but they are present in high prevalence in the infant gut microbiota. At the third month, 100% almost at this sulfate reducing bacteria that can use lactate. So interesting observation in adult associated with disease, but shows also that infants need to be studied for their own, because they are not occurring to the adult. And we'll see afterwards that the levels were surprisingly high. We did also find some lactate utilizing bacteria, but this time not SRB, in all infants from the very beginning, essentially, of the first 2 weeks. Then on the qPCR genus, we could also detect and quantify this time the different population, and confirm also, but more precisely at the genus level, what we observed in terms of functional group by culture. So we did this enumeration, and we found indeed that for the lactate utilizing bacteria, which are not SRB, we detected Veillonella, and this is quantitative by PCR. And you see SRB, they are about 10 to the 5, with some variation among the infants. So they reach high level already after 2 weeks, and the butyrate producer is Anaerobutyricum allia. It changed name also. Before it was called a Bacterium allia, and it's a major butyrate producer in the adult, and it's present only in 20% of the infant. And one of the problems of this bacteria, it's a good bacteria in adult, but it produces hydrogen. It produces gas as well. Okay, so we compared the community of colicky with healthy infant at the level of the lactate utilizer, and this is with PCR. So this is quantitative. It's difficult to do with sequencing. So what we found essentially is that in colicky baby, around 1 month and 3 months of life at the time where colic appeared. And then afterwards, colic, it's only a short time, it's 1 month maybe, or sometimes it can be slightly more, but it's between 1 and 3 months of age. And afterwards, it's naturally resolved. What we saw here is that the lactate utilizing bacteria ratio, which are non-SRB to SRB, are increased in infant colics. So that it would suggest that, the SRB indeed are important for the dissimulation of the lactate. We also observed that there is also a relation between Veillonella and E. alea, and which are producing H2 and the SRB, which are consuming the hydrogen of the system. So we did a different analysis. I don't want to spend time here, but we could confirm that there is indeed a cross-feeding of hydrogen from the Veillonella, from the E. alea. We showed significantly only for E. alea, and we could also show a significant cross-feeding between lactate producers, certain type of lactate producers, such as between Staphylococci and Bacterium alea, and lactate utilizers. Okay, so it kind of provided some support for the etiology of colics, or the reason of colics, related to the overproduction of hydrogen. And hydrogen, it's a problem in adult as well, because it represses butyrate production hydrogen. So it's kind of a regulator in the gut. In infants, there is not much butyrate. It doesn't have this action, but it will create bloating. So what we did, we continued after 6 to 24 months. I don't want to talk about that because it's reported in a very recent paper, and essentially it has been presented very nicely by Catherine. And so here, more or less, the mechanism which is demonstrated on the lactate and the consequence of accumulation, the consequence of transformation by certain microbe to hydrogen, and as well as the consequence of consumption of hydrogen to H2S, leading to possible also damage to the gut of the infant. So we looked for microbes that we could use in order to substitute to the hydrogen producers that produce propionate, so that we don't accumulate hydrogen, and which eventually could compete with SRB, because SRB, we don't want to promote SRB. This is not a way to do. So we focused on propionibacteria. We showed in this first study that and it has been shown by others that propionibacteria are present in the infant gut. You can see the levels is about 10 to the, I mean, there is a big range always, but it's around 10 to the 7 colony-forming or cells per gram of feces. This was by qPCR. And we tried to search. At this time, they were still called propionibacteria. We tried to search, can we find a healthy propionibacteria? Okay, so we did isolation on a number of babies. We found only Cutibacterium avidum, which is the new subcategory of propionibacterium from the skin. And even though we found them, we wanted to try to test our hypothesis of propionibacteria being able to compete with in order to reduce hydrogen production. That's what we did. So we did it in a colonic model. So we developed a very special colonic models that we apply for different hosts, different conditions as well. And I think we are the only one being able to implement a very young age. We need infant from 2 months, or 1 to 3 months. Very hard to get feces at this age. We can only get minutes amount of feces. So we have developed a technology which involves recultivating the system in immobilized form. So we create biofilm in fermenters, which under the set operating conditions, which are very well selected according to those, can regrow the complete microbiota. And so we have a functional colon of an infant. Here it's a 2-month infant, which allows afterwards to test for different treatments. So we did show that there is capacity of colonization of Cutibacterium, and we tested also Propionibacterium strain that came from our cattle culture collection. We showed also the competition for lactate between lactate utilizing bacteria, SRB, and other lactate utilizing bacteria, and also impacting on nitrogen production. We showed the cross-feeding. We demonstrated the effect of pH and retention time. I don't want to explain this. And showed also the competition of mainly the Cutibacterium and Propionibacterium strain that we selected for this purpose. So the last stage was done in vivo. I won't say too much as well. So we bound with a team in France, in INRA, specialized in IBS, and they develop rat models, for IBS, for bloating, and so on. But they are adult rats, so we needed to validate a rat model for infant microbiota. So we validated with transplantation of infant microbiota, quite a challenging experiment. Set up the condition of the diet of the rat so that it can feed on formula. So we needed to add a few fibers because otherwise the rat would explode. And then we tested the composition of the rat in the feces and upon the sacrifice with an intervention which was done with Cutibacterium avidum. At this time we had only this strain available, and what we showed, so we measured essentially H2 excretion in respiration chamber, and this was all done in notobiotic system. So it was very, very challenging experiment, but we showed that when we treated essentially with P. avidum, it's P279, it's the number of the strain, but we see a significant reduction of hydrogen. Very important is that the rats with infant microbiota, they were essentially excreting a huge amount of hydrogen. In normal adult, I think it's about 20 to 50 ppm. Here we had excretion of up to 4,000 ppm. So it shows really very well that the infant microbiota is designed to produce H2. And this H2 indeed is not comfortable for the host. So when we added the Cutibacterium, we reduced by about 1/3 the hydrogen production. So showing that there is the mechanism is holding. It doesn't show that we can resolve colic, but at least we can act on the production of H2. So what the next step? So the next step, it was to try to develop it into a product, and then to do real studies with humans from the academic concept to the discovery phase, and this is done with Vanessa Rocha. We did the PhD on the rat and Cutibacterium experiment, who took the challenge to try to set a startup with a productive mind, combining a well-chosen Propionibacterium. Needs to be competitive in infant microbiota, and for this we needed to test it in vitro model to check the capacity to do this colonization, and we combined it with the Bifidobacterium. So I won't talk about why we did that. It's under patent as well, but hoping to develop a product that we could validate in vivo for both the prevention and maybe the treatment of colics, so that parents can have a good evening during the first 3 months of life. So with this, I'm done. What I would like to state in the summary is that viable microbiology is not a dead science. I'm a bit ironic on this, but I think there is a lot of demand in the microbiology. We need to understand mechanisms. We need to investigate at multiple scale, from in vitro reactors to models, and afterwards, obviously, to animals and to humans. The study, I think, is still the first mechanistic study of infant colic with microbe function. And we also revealed taxonomic and functional transition in the system based on lactate and hydrogen. And finally, this illustrates also the window of opportunity to support dietary intervention with functional characterization for promoting health, in particular in the infant in this case. So I would like to finish by acknowledging on this slide, I mean, we had very many people involved over the 12, 13 months. I put only I would say the most important. I would like to also to highlight the three study nurse. There was a study doctor, the first one, Daniela Rogler, who supported tremendously because to get fecal sample from the time of delivery, so this was outlined almost days and nights, and have the laboratory ready with all the method to be able to treat the sample within a couple of hours. This was a tremendous challenge and providing complete anaerobic conditions. And so with this, I would like to close my presentation. Thank you for your patience, and I wish you a good coffee break now, and also continuation with the next speakers.
Thank you very much, Christoph, for a really nice presentation about the work of 15 years. I think we are a bit tight in time, so I think we have time for two questions to Catherine Stanton and to Christoph Lacroix.
Maybe to Catherine first, if you agree.
Any questions to Catherine?
You don't get online question, eh?
No.
No, it's not. Okay.
Or to Christoph, any comments? Or yes, please. Yes. Yes, I have to bring you the microphone. Just hold on, please.
So you said that perturbation of the microbiome could potentially lead to obesity. Is there any is this causation or correlation? Is there any mechanism by which perturbation of the microbiome can lead to obesity?
It's not clear, I suppose, mechanistically. The altered microbiome would have altered metabolic functional capabilities with altered ability to harvest energy from the diet of the host, and that would promote potentially adiposity and ultimately obesity. So that's one possible mechanism. It's a good question, but I think more studies are required, and it's very difficult because in the human host, I mean, you're talking about long-term studies and really only observational studies to try and investigate it, and then do some sorts of model studies to prove it. But I think energy harvest from the host has certainly been one area that has been looked at and concluded that it has a potential to impact on energy harvest and adiposity accumulation. Thank you.
I have a question in a similar vein. Brain or gut-brain axis. So what is the mechanism actually to the brain? It's not only correlational evidence, but it's how can one explain that there is an impact on modernization and things like this?
It's a very complicated question, I think, and a very complicated topic. Certainly, the gut microbiome produces a lot of compounds that are potential, or some of which have been shown to be neurotransmitters and impact on some aspects of brain function. So the altered microbiome associated with C-section or antibiotic exposure, whatever, certainly could impact on that and the metabolome. But there are other aspects that are much more complicated and require a lot more work in order to answer that question properly.
I have another question to Christoph. The classical crying curve is, as you said, a peak at 6 weeks and then it vanishes up to 12 weeks. But another interesting feature is that crying increases in the course of the day. So how can you explain that diurnal change? Is hydroxygen increasing in the course of the day, or what is the explanation?
Yeah, there is also a cycle of the microbiota, but I mean, again, according to our mechanism, hydrogen is produced from the dissimilation of lactate. Lactate is produced from the consumption of human or milk or of formula, and babies usually eat more, drink more in the day. So that could be a potential for accumulation of sugars in the gut and then feeding the chain. But again, it's a very good question. It has also, it could be a mechanism, but the sensitivity also of the gut in the infant might be increased at the end of the day when you have more stress, more tension, more psychoactive environment in the infant, tired parents as well. So it's a good question, definitely.
The last question, Carsten.
Regarding health and disease and the microbiota, I think we have to study not only the feces, but also the microbiota which gets into interaction with the epithelial layer and the immune system, which mediates the signals. So if you mentioned Crohn's and other inflammatory bowel diseases, I think it's very necessary that we study what is happening on the barrier side. And I think it's also necessary to study the other bacteria, which factors they produce, but I think we have to dissect the microbiota on the epithelial layer and the microbiota from the metabolic side. So that is just a comment, and maybe you can comment on this.
Absolutely, you're totally right, of course. And I mean, the fecal analysis isn't ideal, but it's an easily obtained sample, of course, and that helps a lot. But certainly obtaining samples via various procedures in the gut, in the right place, where the activity is happening, is crucial to ultimately understanding the impact of how the microbes interact with the mucus layer and the intestinal epithelial layer and all the functionality associated with that. There are tools now that are useful for sampling along the gut, including various types of capsules that are being developed, which hopefully will give a lot of insight into the future. And also, I suppose, animal models have their limitations. We all agree with that. But I think the pig model might be a potentially more closely anatomical model relevant to the human gastrointestinal tract, and might have further potential to give some further insight into that. But you're right, that's absolutely where we need to go with this. Last word.
Thank you. Yeah, this is completely right. You have both a longitudinal gradient and an axial gradient in the gut, but this is very difficult to approach in reality. When we talk about metabolites, I would say most of the metabolites, they are produced by the bulk, and the bulk is in the luminal part of the intestine. So if you might have also bacteria associated with mucus based on metabolites amounts, probably it's small, but it can still be very, very active. And you have also a component of the microbial cells which interact directly with the immune system, which might not occur in the lumen, but will occur in also in the mucus. So it's part of a very complex system, which is very difficult to approach. It's my humble opinion on this.
Thank you very much for your attendance, and thank you for the presentations and the discussions. We have now a coffee break. I would suggest that we make it until 25 instead of 15. I think the best is if you follow Vanessa. She knows the way. It's in the alumni pavilion. So you have to walk probably about 2 or 3 minutes and then come back here for the next 2 presentations. Thank you.